This section is about glucocorticoids. We'll discuss mechanism of anti-inflammatory and immunosuppressive effects of glucocorticoids. Then we'll discuss anti-inflammatory effects of glucocorticoids on different aspects of inflammation and also immunosuppressive effects of glucocorticoids. And finally, we'll discuss risks of infections associated with their use. Before we start, we know glucocorticoids are used for treatment of various conditions for their anti-inflammatory and immunosuppressive properties. Cortisol is a natural glucocorticoid and it is main regulator of stress response. And it also has anti-inflammatory and immunosuppressive properties that help to prevent hyperactivation of inflammatory and immune response. Similarly, synthetic glucocorticoids also have immunosuppressive and anti-inflammatory properties, and synthetic glucocorticoids also have metabolic effects that contribute to side effects of these drugs. Now let's discuss mechanism of action of glucocorticoids. Glucocorticoids have their effects by two mechanisms. One is genomic effects, that is due to effects of glucocorticoids on transcription of different genes, and second is non-genomic effects that do not involve transcription of genes. Most effects of glucocorticoids are due to their genomic effects. First, let's discuss genomic effects. Glucocorticoids are steroid hormones, so they can easily cross cell membrane due to their lipid solubility. Glucocorticoids then bind to glucocorticoid receptors present in cytoplasm. Binding of glucocorticoid to glucocorticoid receptor releases glucocorticoid receptor from associated proteins. Hormone receptor complex then moves to nucleus. In nucleus, hormone receptor complex affects transcription of genes by three mechanisms. First mechanism is in which glucocorticoid receptors directly bind to DNA, and they bind to site of DNA called glucocorticoid response elements or GRE. Binding of glucocorticoid receptors to this part of DNA results in increased expression of some genes. Expression of which genes is increased by this way that are genes that contribute to metabolic effects of glucocorticoids, like genes related to gluconeogenesis and genes that contribute to anti-inflammatory effects of glucocorticoids like IL-10 and lipocortin 1. This is called transactivation. And like GRE, there are other sites on DNA that are called negative GRE or NGRE. And when glucocorticoid receptors bind to these sites, it suppresses transcription of genes. And this is called transrepression. So in GRE and negative GRE, glucocorticoid receptors directly bind to DNA. In contrast, in tethering, glucocorticoid receptors do not directly bind to DNA. Instead, they bind to a transcription factor that is already bound to DNA and inactivate this transcription factor. And this transcription factor then cannot increase expression of genes. And this is one of main mechanisms by which glucocorticoids inhibit expression of genes that contribute to inflammation. So tethering affects transcription factors like NF-kappa B, AP1, and NFAT. Third mechanism is called composite method. In this method, glucocorticoid receptors have two effects. One is glucocorticoid receptors bind to DNA and increase expression of some genes. And same glucocorticoid receptor interact with adjacent transcription factor and inactivate it. Now let's see which genes are inhibited by glucocorticoids and which genes are activated. Before that, we briefly review inflammatory response and see how glucocorticoids exert their anti-inflammatory effects. Cell surface receptors like toll-like receptors detect pathogen molecules and damage signals, and they send downstream signals that result in activation of transcription factors like NF-kappa B, and these transcription factors then increase expression of some genes that results in production of mediators of inflammation like prostaglandin, leukotrienes, and cytokines. These mediators then contribute to inflammatory response and result in vasodilation, increased vascular permeability, and infiltration of inflammatory cells. Glucocorticoids can affect transmission of signals from cell surface receptors. They can inhibit transcription factors like NF-kappa B, and they can decrease production of inflammatory enzymes that produce prostaglandins and leukotrienes, and they can decrease production of cytokines. And these mechanisms affect different phases of inflammation, and this is how glucocorticoids contribute to anti-inflammatory effects. We can see how inhibition of a transcription factor can affect innate and adaptive immune response. NF-kappa B is main regulator of inflammation, and it results in production of cytokines, chemoattractant molecules, cell adhesion molecules that contribute to neutrophil infiltration, and it also increases production of inflammatory enzymes like cyclooxygenase. So when this transcription factor is inhibited by glucocorticoids, it decreases production of all these mediators and it significantly affects inflammatory response. Similarly, this transcription factor also contributes to activation and proliferation of B and T lymphocytes. So inhibition of NF-kappa B by glucocorticoids can also significantly affect adaptive immune response and contribute to immunosuppressive effects of glucocorticoids. Now let's see which genes are inhibited by glucocorticoids and which genes are activated. Glucocorticoids decrease transcription of pro-inflammatory genes. These are genes for cytokines, chemoattractant molecules, cell adhesion molecules, and inflammatory enzymes. And glucocorticoids increase expression of anti-inflammatory genes. These are genes for IL-10, lipocortin, NF-kappa B inhibitor. So combination of decreased transcription of pro-inflammatory genes and increased transcription of anti-inflammatory gene contributes to anti-inflammatory effects of glucocorticoids. Many of these genes are overexpressed in chronic inflammatory conditions, so treatment with glucocorticoids provide adequate anti-inflammatory effects. However, prolonged use of glucocorticoids is associated with significant side effects. Glucocorticoids also decrease production of cytokines that promote inflammation, like IL-1, IL-2, IL-6, TNF-alpha, and interferon gamma. And many of these cytokines also contribute to adaptive immune response. In addition, glucocorticoids also decrease expression of co-stimulatory molecules on antigen-presenting cells and lymphocytes like B7 and CTLA4, and this also affects adaptive immune response. So we have discussed genomic effects of glucocorticoids, and genomic effects take time to occur as they result from effects on transcription of genes. In contrast, non-genomic effects can occur within minutes. Non-genomic effects of glucocorticoid occur due to interaction of glucocorticoids with glucocorticoid receptors. These receptors are present on T cell receptors and on cell membrane, and glucocorticoid receptors are also present in cytoplasm. Activation of glucocorticoid receptors on T cell receptors affects transmission of signals from T cell receptors, and activation of cell membrane receptors affect functions of other proteins in cytoplasm. High-dose steroids can interact with cell membrane and can alter structure of cell membrane. This affects transmission of ions through cell membrane and can significantly affect functions of cells. And this can result in profound anti-inflammatory and immunosuppressive effects. Glucocorticoid receptors are also present in cytoplasm, and these receptors can interact and inhibit other proteins like that of MAPK pathway. And these receptors can also translocate to nucleus and result in production of inhibitors of MAPK pathway. And MAPK pathway can also be inhibited from signals from cell membrane receptors. So there are several ways by which glucocorticoids can inhibit a particular pathway. So glucocorticoids have their effects through genomic and non-genomic effects. And synthetic glucocorticoids bind to same receptors in cytoplasm like cortisol. However, synthetic glucocorticoids bind to these receptors with higher affinity, so they have more potent glucocorticoid effects. However, synthetic glucocorticoids bind to mineralocorticoid receptors with lower affinity, so they have less potent mineralocorticoid effects. So in this section, we discussed about mechanism of action of glucocorticoids, and in next section, we'll discuss about anti-inflammatory effects of glucocorticoids on different aspects of inflammation.